All right, so I'm going to go over the slides. These are hyperlinked, or this video is hyperlinked in 21 to 26 for your notebook. So I'm going to go over that. Occasionally, I'll press pause, or not press pause, sorry. I'll ask you to pause if you are doing stuff in your notebook. So please remember to pause the video at that time. Otherwise, the video is just going to continue and you're not going to be able to keep up super fast. All right, so we're talking about a disease, okay? So in our notebook, we have a description of a patient. So this patient comes in, he's got some different symptoms. These symptoms include things like fatigue and et cetera, et cetera. What I want you to do is read through the patient description, and you're going to write down in your notebook what you consider the most useful clues to help you figure out the disease. So this could vary person to person, obviously, but I want you to write down what you think if you were his doctor, what might start pointing you in the right direction. In your notebook, go ahead and list these as bullet points, and you also don't need to have a reason written down, but mentally I want you to think, okay, this is why I think that clue is useful. After you write that list down, I want you to tell me what kinds of tests you could do to determine what disease these symptoms represent. All right, so if you are doing your notebook page, go ahead and press pause now. All right, so useful clues. You might have included some things like his symptoms. You also might have included the fact that it seemed to afflict other family members. Sometimes students find it helpful that he's from a specific area of the world. Some people don't write that down as a clue. Some tests that you guys tend to tell me about are things like x-rays, blood tests, urine tests. Some of you mentioned MRI or CT scans. But one pretty consistent test that you guys mentioned is a blood test. All right, so they actually did do a blood test. You have pictures of the patient's blood samples. You also have a picture of a normal, unafflicted patient. So this blood sample on the left is someone without the symptoms, and on the right is the patient. You have, again, these in your notebook. So I want you to write down what evidence you see in his blood sample that might explain his symptoms. Okay, go ahead and pause the video, complete that, and come back. All right, so you should see that this guy seems to have a lot more blood cells. That's one big clue, right? And then another clue potentially is we've got these weird shaped ones going on here. So that it doesn't seem right versus this guy where they're all round. Some people might mention the color. This actually is just kind of due to, they use different, what they're called stains to make the slides. So they're showing slightly different colors. All right, so now I'm going to show you the mini activity. To do this mini activity, you'll need a tube and some Play-Doh or clay. It doesn't have to be pink, but I'm using pink to represent blood cells. And you're going to make some blood cells. You are going to make round blood cells. It says 10 to 15. You don't need to make that many. And we're going to make some of these kind of crescent-y, misshapen ones to represent those blood cells as well. And then we're going to drop the tube, not the tube, we're going to drop the blood cells through our tube. So I'm going to switch my screen so you guys can see what's going on. All right, so what we've got here is a paper tube, okay? So this paper tube is just a piece of paper with tape, and it's hollow, okay? Then I've got my Play-Doh. I know it looks orange on your screen. It is not, it is pink. So I'm going to make some blood cells. Now, blood cells need to be able to fit into the tube they're being transported in, right? So I need to make sure I'm making my circles small enough to get through the tube. So that's about right. Again, these are representing our blood cells. This is our blood vessel, like a vein or a capillary or an artery. All right, so normally what happens when I put a blood cell into the blood vessel? It just goes right through. There it goes. Okay, they're going right through. Now, we had our patient, and he had some misshapen cells. So he had some weird, so I'm using about the same amount of clay that I used to make, you'll see, this guy. So I'm going to misshape one of them because he had some weird shapes. He had kind of like a crescent moon shape. And then he also kind of had like this flatter one. So I'm going to make kind of like a flatter one. And what happens when I put those guys in? Okay, so if we need to repeat, the regular guys go in. What happens when I put my crescent guy in? Well, he won't even go in. Okay, he went in eventually, right? But he kind of went slow. Let's see if we try again. Let me just try and make sure I've got it on the screen. Push, come on, you got this. He's getting stuck. I have to like push him through, okay? Let's try the other weird misshapen one. Oh, he went through pretty good. Good, okay. Uh-oh, we're stuck again. So, he keeps getting stuck, right? And I keep having to like push him through. Keeps getting stuck. Let's see if we can get him in there a little better. Okay, I have to like push and deform him. Okay, so if you didn't do the mini activity, go ahead and write down your observations. If you did do the mini activity, go ahead and do your observations based on your own activity. Okay, so you should have described the results in your notebook. And what we saw in my kind of little mini activity is the misshapen cells seem to be getting stuck, right at the beginning especially. I kind of had to like push them in. So how do you think that might relate to some of his symptoms? So go ahead and answer that in your notebooks, press pause, and come back. All right, so if we have our blood cells getting stuck, this might start to explain some of the symptoms like the pain the guy is feeling, or maybe some of the swelling he's experiencing, okay? We're gonna find out that it actually connects to quite a few of his symptoms in a few slides. And if you remember, blood cells deliver oxygen. So remember, if we go back, he's feeling fatigue. And so if his blood cells are getting stuck somewhere, he's not getting the oxygen he needs. So he's going to feel tired. And if you remember last year, oxygen is a key component in making ATP, or the molecule that cells use for their energy. And so if his cells aren't getting enough oxygen, they're also not going to get enough energy. All right, so this is kind of what you should have seen happening, right? Normally, we get the flow nice and through. With people who have the misshapen cells, we're getting some blockages, and that's not good. All right, so our disease finally is named. It's called sickle cell anemia. They name it after the blood cell shape, which is kind of like this sickle or crescent moon. This is a sickle. It's a garden implement. Dr. Ernest Iron, he is the one who ended up naming it. So you can see this is what a normal blood cell should look like. This is our sickle cell. As you can see, that is not round, and it can get stuck in the blood vessels. All right, so he had low energy, trouble breathing, general achiness. These can all be attributed to his lower oxygen levels. So those blood cells that are misshapen, we don't know if they're carrying oxygen as well at this point. We do know they're getting stuck through our little model, though. So we know he's not getting enough oxygen potentially to his cells. So if you have low energy, that again connects to cellular respiration. Trouble breathing. So when your body is lower in oxygen, a lot of times your body's going to send signals that you need to breathe more. Even if you have enough oxygen in your blood, your body, if your body's not able to get it and use it, it's going to say, hey, breathe more. And that might cause some things like hyperventilating, etc. And then general achiness, typically when you have lower oxygen levels, your body is going to switch to something called fermentation or lactic acid respiration, and it's going to build up some lactic acid, and that kind of causes like a burning, achy sensation. You might feel this if you've ever done like a plank for a super, super long time or something, or a wall sit. Your muscles start to burn, and this is going to be due to switching which type of respiration they're using. We also noticed that in his picture, he had fewer cells, and later studies show sickle cells die faster than the average red blood cell. So your average red blood cell sticks around for about 120 days. And our sickle cells are dying in 10 to 20 days. So if you think back to his pictures, he had fewer cells, and that kind of makes sense if his blood cells are dying more quickly. In the 1920s, in the late 1920s, two new doctors, Dr. Hahn and Dr. Gillespie, discovered that the regular blood cells from the patient could turn into those sickle cells in low oxygen conditions. So if you remember, our patient, he felt really, really tired after swimming or exercising. And this kind of connects to that. It's showing that maybe his blood cells are kind of normal if he's just doing normal things. And then in low oxygen conditions, like when you're working out and your body doesn't have as much oxygen as normal, maybe, or if the blood cells have to move more quickly and maybe are getting stuck more, they are sickling more. And so you see his symptoms get worse. He also has some pain and swelling. Again, they're getting stuck, so that could connect to our pain and swelling. He also had chest and abdominal pain. And so what's happening here is his cells are getting stuck near the lungs in particular. This is just one of the common places they get stuck. They can also pool in the spleen. So your spleen is there to help kind of like recycle old blood cells, so they travel there quite often. And they break down and recycle parts. And so when they get stuck there, it causes the pain that he's feeling in his abdomen. And then yellow skin and eyes, a lot of you mentioned yellow fever or have in the past, or things like hepatitis or jaundice. So your liver actually gets rid of dead blood cells. They also get rid of toxins. And if you have liver damage, for example, in like yellow fever, your liver can't get rid of toxins, so you start to look yellow. In this case, the toxins from the dead cells that are dying really quickly, the 10 to 20 day life cycle they have makes them die. They have toxins in there that the liver generally can get rid of, but they're dying quicker than the liver can process them. So you end up kind of getting this yellow, sickly skin and eye color. And by the eyes, I mean the white of the eyes, not the pupil and iris. All right, so what the heck is happening in this blood cell? So it's actually going to be related to a protein. So there's a protein in your red blood cells called hemoglobin. Normal hemoglobin in a red blood cell looks like this. It's designed to carry oxygen, okay? And then in the 1950s, a scientist by the name of Linus Pauling, he discovered that this protein in people who have sickle cell, instead of looking like this, it looks like this. And this is super long and inflexible, which means in their blood cell, you can see that would fit nicely right in the middle there. This can't fit in that round shape. And so it causes the whole cell to become this misshapen shape. And it's inflexible, meaning when it goes into the blood cell, it's rigid. The blood vessel, it's rigid and can't like bend and easily pass through. And so it's stiff, they're angular, they're getting stuck here, okay? This is the first time we connected any disease to an abnormal protein in the body. We have since discovered quite a few different things, but this is the first one. We've got a protein that is going wrong. All right, so in mini activity 2, you're going to go into your notebook for this one. You're going to go to this slide here. For mini activity 2, that's 3. 2, you're going to use your codon chart to figure out what amino acids are being made. Okay, and then I will go over some stuff with you. So if you could pause here and do mini activity 2. And your codon chart is in your notebook. So make sure you're using your codon chart. And there's an additional video if you would like to watch after I go over mutations. There's an additional video for you there. Go ahead and press pause. All right, so you're back. So you should have seen that there were something called a mutation happening. And these are mutations are changes to the DNA. So right there. Mutations aren't necessarily bad. They're not necessarily good. They could be bad or good, or they just could be neither. So if we go back to your mini activity here, what you'll see is we have this mutation and it's had a base added. Okay, so we added the C there. This should have thrown off your entire sequence of amino acids compared to this one up here. Okay, in this one, where one of the bases gets deleted, the same thing's going to happen. It's going to throw off your amino acids. So as soon as that deletion happens, everything after that is like a domino effect. Okay, and in this one, where we had one of the bases replaced, it should not have had as big of an effect. It probably just affected one amino acid, potentially two. Now, what's really cool is if the amino acid on the codon chart, if the change coded for the same amino acid, you wouldn't even notice the mutation at all. So sometimes mutations don't get noticed. And again, this is kind of review if we went back to your cow slide here. Remember when you were doing this one, we had a change that occurred that didn't affect the protein. Okay, so again, there's 3 types of mutations that you should have just seen here. You should have seen when we add a base, shifts everything down. When we delete a base, shifts everything down. And a replacement typically affects less, okay? Now, some mutations are super harmful. Some mutations are helpful, and we'll go over that. And then some, like this one, may not have any effect at all. But a mutation is just a change in the DNA, okay? So if we go back to mini activity 2, all right, so mutations can lead to non-functioning or damaged proteins. So in the case of adding in or deleting a base, a lot of times that's going to result in a non-functioning protein because you're shifting everything in the chain. It's like a domino effect. In the case of replacing a base, a lot of times those could damage the protein or be what we call silent, where you don't see any change at all. And then some mutations happen and they actually end up being beneficial, okay? So for example, back here, we've got the ancestor of all the bears around 30 million years ago. And what's happening is mutations happen in our DNA all the time. And when we get a mutation that's helpful, you often see splits in the population. So for example, polar bears and brown bears, they come from the same ancestor species here. And at some point, there was a mutation in some of them to get white fur. And this actually turned out to be a beneficial mutation. Actually, their fur is clear, not white, but this turned out to be a beneficial mutation for these bears who lived in the Arctic region. And so this mutation was helpful. So occasionally that change results in a change that gives you a more chance of surviving wherever the heck you are, okay? All right, so we're gonna do mini activity 3. So we know we have an abnormal protein, so that means we probably have a mutation. So I want you to pause the video, go into your notebook, and complete mini activity 3. So pause now. All right, so mini activity 3. We've got our normal hemoglobin here. We've got sickle cell anemia DNA here. This is the actual DNA that we see in people who produce normal hemoglobin and people who do not. So you'll notice you should have done your RNA and you should have done your amino acids. Okay, so you can see we got CET, CTG, CCT, GAG, blah, blah, blah. There's one small difference that you should have highlighted in your notebook. The difference is right in this one here. Instead of CTC, like you and I, if you don't have sickle cell, we would have this one. People with sickle cell right there have this A. They have one change to their DNA, and that is it. This disease is caused by one base being different. So instead of having CTC, their DNA has CAC, and it changes the amino acid, and that now is a non-functional protein. And now they have sickle cell anemia. All right, so at this point, you should have made it through your notebook slides. If not, make sure you go back and do all the associated work with this video.